Neha Mishra. I am a postdoctoral researcher working with the EU-funded project SYSCID. I am a bioinformatician by training, so I analyze biological data for a living. I work at the Institute of Clinical Molecular Biology at the Kiel University. So within SYSCID, we are interested in identifying the molecular signatures of chronic inflammatory diseases. So these are the diseases which are first chronic. So once the patient has it, the likelihood is that they are going to have it for the rest of their life. And this is caused by when your immune system treats things that are not supposed to be as pathogens, but it treats it like a pathogen, causing inflammation when you don't have it. So a good example is, of course, inflammatory bowel disease, which is the disease of the gut or the digestive system, or you can have rheumatoid arthritis, in which your joints can become inflamed and so on. What we do is we profile a lot of molecular data from the patients from these diseases, and this could be RNA sequencing data, DNA methylation data. So we look at epigenetic marks of the patients, or genotyping data, which is like the genetic information from the patient. And this is like a huge amount of data. So my role within SYSCID is to analyze this data and use this data to answer the questions that we have defined. So for example, what is the molecular signature of inflammation in general across chronic inflammatory diseases, or what happens when a patient is given a therapy, and are there any markers in this kind of data that can tell us whether the patient is going to respond to that therapy or not. Because you have to keep in mind that these therapies are not very easy to give. They also have some side effects and high costs, and many of the patients, approximately half of them, do not respond to it. So it would be very nice if we could know, very efficient and very good for the patients themselves, because they don't go through the process of having a medicine that's not going to work for them in the future. So within SYSCID, I'm involved in a project for therapy response in IBD, so that's inflammatory bowel disease, in which we are interested in finding some molecular signatures that we can find before or in the early stages of the therapy that could predict whether or not the patient is going to respond later on. And one of the major findings that we have from this project is it's not only how the gene expression is before the therapy, but also how the initial response to the therapy is between patients that can determine whether or not they are going to respond in the future. So this was a major finding, and we found gene expression markers as well as DNA methylation markers, which means the epigenetic changes that occur after the therapy is given, that were predictive of the therapy response later on. In case of inflammatory bowel disease, it is quite important, as in other inflammatory diseases, that we have some kind of therapy that is tailored to different patients that would work, that would actually work for them. And there have been quite a number of studies that are, including ours, that are looking into that. One of the major challenges that I feel would be the applicability of the things that you find in the research in a real clinical setting, because that could be quite a difference. Because when we do the research, we profile as much as we can. So we do the whole transcriptome, we do the whole genome, we do the methylome and so on. But we can't do that. It's not practical to do that for every patient that comes into the clinic. So we want to find markers that are fewer and precise that can help in determining. And the other thing that I feel that is a major challenge in precision therapy is, of course, when we do research, we do it on the local population. For example, when we are in Europe, we are doing it in the European population. A person who would be doing that in China would be doing it in the Asian population. And of course, then there are other countries which don't have enough resources to do that altogether, which have a different ancestry altogether. Now, the important thing would be how these signatures are, to compare how these signatures are between populations, and whether what we find in the European population can be applied to someone, for example, who is an Indian person or not. So I think that's one of the major challenges, not just for IBD, but all chronic inflammatory diseases. The most exciting aspect is the research itself. Given that these projects are so interesting and so relevant, that I do get to try a lot of new ideas, try a new analysis and stuff, that's the exciting part. And again, networking, that we get a chance to do that, is also very exciting. The challenging part comes also with the exciting part, because as I said before, finding something that is relevant from the whole profile of a patient. It's like finding a needle in a haystack problem, and you have to think of always new ways to find not only like specific markers, but also the ones that can be replicated or validated in other studies, in other cohorts. So that's kind of sometimes stressful, but most of the times very a good opportunity to try on new analysis and basically improve your whole profile as a scientist. Like the more you do, the more you learn, and the more then you have to offer. So I think participating in EU funded projects is a good opportunity for the young scientists because they get a lot of exposure and networking opportunities. The main thing is the big collaboration that you have with other partners, and then you get a fresh perspective on your work from the people who are not from your group. And like having meetings like these where we get to meet each other, it's quite exciting because we get to discuss more science in an informal way, get opinions from other professors or even other colleagues in other groups, and just a great networking opportunity that we get here. For young scientists, this is especially important because they can also have a chance to look into what other labs are doing, and they might want to work with them in the future. So it's really good that you know now many other people who are working in the field.